Welcome to the SIBO Doctor Podcast with Dr Dr Jacobi. If you're a practitioner and want to learn the basics of SIBO, head over to sibotest.com and sign up as a practitioner. This will give you access to a 90-minute webinar on the fundamentals of SIBO treatment. If you're a patient, please know this information is not intended to diagnose or treat a medical condition. Please ask your doctor before initiating any new treatments. And now, over to Dr. Jacobi and the SIBO Doctor Podcast.
If you're like me, you're finding that treating small intestinal bacterial overgrowth can be complicated. Patients often present with a myriad of symptoms and associated conditions which require holistic thinking and a methodical approach to treatment. This podcast was born out of a desire to start a conversation with other practitioners, educators, experts, and researchers, and see if their insights can give us new treatment considerations, especially for our difficult cases. In this podcast series, our guests talk about functional gut disorders, hormonal issues, the enteric nervous system, food intolerances, the immune system, the microbiome, methylation and genomic issues, and everything else that can make a SIBO case very confusing. I believe that by understanding the often complex interconnection of SIBO with other disease manifestations, we can be more effective in our treatment approaches. I hope you find this podcast useful in your practice, and thank you for listening.
Build your clinical confidence as a SIBO treating practitioner. The SIBO Masterclass is now available at sibotest.com. Presented by Dr. Jacobi, this masterclass covers a wide array of topics in advanced SIBO case management. Learn about implementing Dr. Jacobi's biphasic treatment approach, in-depth herbal protocols, how to manage food intolerances such as salicylates and histamines, methylation and nutrigenomics in chronic gut cases, and much, much more. This course is presented over 4 modules and is available for immediate download. Enroll today and download this incredible tool so you can help even your toughest SIBO cases today.
Welcome, SIBO practitioners. Today, I'm speaking with Dr. Denise Furness, a molecular geneticist, registered nutritionist, and registered exercise professional. She conducted her PhD at CSIRO Human Nutrition before completing a postdoctoral fellowship with the University of Adelaide's Research Centre for Reproductive Health. Specifically, her research investigated folate, nutrigenomics, methylation, and DNA damage in relation to pregnancy outcomes. She has published her work in peer-reviewed medical journals and regularly presents at national and international conferences. In 2012, Denise shifted from an academic research position to a consulting role at Your Genes and Nutrition, helping patients and practitioners understand the role of nutrigenomics and genetic testing in relation to various health outcomes. In particular, Denise is passionate about helping couples achieve optimal health and wellness to improve their chances for natural conception and healthy pregnancy outcomes. In addition, she holds regular education seminars and workshops discussing the links between our genes and our environment and how these impact on our health throughout all stages of the life cycle. So a very warm welcome, Dr. Denise Furness.
Thank you. I'm looking forward to chatting with you, and thank you for that lovely introduction.
So I met you actually just recently at the Bioceuticals Research Symposium, where you gave a really riveting masterclass, if you will, on nutrigenomics and methylation. And I thought it would be so great if we could have this conversation on the SIBO Doctor podcast. I get a lot of questions about the role of nutrigenomics and methylation, because it's a hot topic right now.
Yes.
And so I thought I'd just bring you on the show and sort of pick your brain about just an overview of methylation, just briefly, but also what should we look out for in our patients that have ongoing digestive symptoms, and how can we utilize some of the genomic testing that's available?
So I guess in a nutshell, the reason why there's so much focus on methylation is because it's involved in so many processes in the body. We know that it's important for switching genes off and on, so it has that epigenetic component, and this is a huge has a huge impact on our cells and our body, because turning genes off and on can relate to so many different things. As you mentioned, my work was in pregnancy, so obviously when you've got a developing embryo, you need the right genes switched off and on. Plus, it's linked with detoxification, neurotransmitters, and it does so many things. And this is why we continually hear about methylation. It's important for so many functions. But when we get down to it, it is quite difficult actually to truly measure methylation. And sometimes people say, I'm an over and an under methylator. And I think sometimes these things are simplified. It's not as simple as that. And I want people to know that when I start talking about how to test for methylation, remember, we're not actually testing the methyl groups. So methylation really refers to a methyl group, which is a carbon and 3 hydrogens sitting on top of DNA or a protein or RNA. And then by sitting on the DNA, it can actually impact on how something functions. And to measure these methyl groups on the DNA, for example, and I did that back in my research, we looked at it in placental tissue and the mother's tissues and made some comparisons with pregnancy outcomes. It's truly difficult. So when we're in the clinic, which I am now in the clinic, as you mentioned, we are looking for, markers or things that might give us an indication of methylation. And you asked, why would it be important in digestive issues? Well, I think particularly for detoxification, and we also know about the gut and the brain axes. We know that methylation is needed to break down sort of neurotransmitters like adrenaline and dopamine and things like that. So, and even COMT or more MAO is sort of with the serotonin, and we know that serotonin is so important in the gut. So when we're looking at methylation, there's obviously the genetics, which is what I do, my area of expertise and what I love doing. And if you're happy for me just to keep talking, I'm just talking about...
Please do. I'll jump in with particular questions, because I already know that we'll go into a little bit more detail with COMT and MAO and those kinds of things. But, just wanted to kind of also say that one of the reasons I think we see a lot of this issue arise with chronic digestive disorders, or SIBO in particular, is because there are nutrient deficiencies, and also hydrogen and methane gas are directly damaging to B12 and folic acid. So long-term SIBO can actually sort of bring to the surface an underlying methylation, potentially a methylation issue, but just like you said, it's way more than just the sort of inner sanctum of methylation. There's all these other downstream issues that result from that we often see in practice. So I definitely want to kind of go towards histamine and other things that we often kind of see in practice. Histamine intolerance is a huge issue with chronic digestive issues. So can you keep talking, but
So I guess what I was going to go into was the testing. I just thought I realized I'd been talking for a long time. I do get a bit excited sometimes and just start going off on tangents. I could talk about it all day, as you know, when you saw me at the symposium.
And you talked
3 days. So with the testing, many of you would have heard about MTHFR. And my point of view is that MTHFR is only a very small piece of the puzzle. And I understand that it does have a big impact. So these genetic variations, particularly at the 677 position within it, MTHFR, if you have homozygous for the variant, so two of the variants, by the way, I never use the word mutation. So if you're a practitioner out there, I really urge you to stop using the word mutation, because some patients will go away and that will really put the fear in them, or maybe might have some sort of anxiety for no real reason, because these are just variations. They're not MTHFR scientifically is a mutation because it has a significant impact, but a lot of the other genes that we're going to talk about in this podcast actually aren't true mutations, and sometimes a variation on the odd occasion will have a beneficial impact. So I'm going to use the word variation, and for those of you that use mutation, consider my advice there on using the word genetic variation. So anyway, the variations in MTHFR, can lead to up to a 70% reduction in enzyme activity. And there's so much research. It started back in the 90s. A lot of research and money was looking at homocysteine and MTHFR and cardiovascular disease. And then that moved into the fertility, the work that I was doing. And now there's heaps on mental health and things like that. So it's not surprising we hear so much about MTHFR, but it's just one gene that codes for an enzyme in a really important pathway, that sort of folate metabolic or methylation cycle. So I would say to you, the practitioners out there, if you want to look at this from a genetics perspective, look at the MTHFR, but then look at the genes around it. So MTHFR needs to pass on that methyl group to homocysteine using methionine synthase and methionine synthase reductase, MTR and MTRR. These work together and they are super important in converting homocysteine to methionine, which then links into that S-adenylyl methionine, so directly leading to methylation. And that MTR and MTRR require that methyl B12. So MTRR makes sure B12 is in that methylated form, so it combined with MTR. And as you just mentioned, Narella, the B12 can be an issue for people with gut disorders. And I'm going to be completely honest and say I don't know a lot about SIBO, but I'm so glad I met you because now I'm going to learn more about what you do and we can combine forces. But the B12 is really important. And then there's other genes in the pathway as well. CBS going down the transsulfuration, it might pull homocysteine away from methylation and things like that. So I try to look at things at a bit more of a global perspective. And then exactly what you just mentioned, the nutrients. These things don't work in isolation. We want to talk about the cofactors that bind these enzymes, the things that can support and drive enzyme activity and sort of compensate for some of these genetic variations. So we want to know, what are the folate levels? What are the B12 levels? What are the B2, which binds with MTHFR? What about B6? So when you want to identify methylation issues, you're looking at the genes, which is definitely what I do, but also the nutrients. Obviously, there are other markers too. You might be able to get an idea of what's going on with histamine. But really, too, if you truly believe someone has a methylation issue and you want to delve really deep, go off and get like a methylation profile. There's so many different companies that offer different ones, and you can get actually the various levels of folate, like 5-methyl tetrahydrofolate, or you can get the S-adenylyl methionine versus S-adenylyl homocysteine information. I often say that ratio, that SAM-SAR ratio, I think is truly probably the best indication, because remember I said we're not truly measuring methylation, but that SAM-SAR is probably your best indicator if someone has a methylation issue. So that's the testing stuff.
Okay.
So I've talked about the importance, talked about the testing, and
you're actually before we got started, you said you can talk about what your current project is. Can you tell us a little bit more about that, because that directly ties into what you just said?
Yeah. So for those that know me, you'd know that I'm coming back to work after maternity leave, and I have been presenting at conferences for a while, and I've just started back at the clinic. So anyone wanting to come and see me, I'm back at Brighton in Melbourne at Your Health. But I've also been working for quite a few months sort of overseeing a project with bioceuticals. So some of you out there might know about it, but we have been trying to keep it under wraps. And what bioceuticals are moving into is clinical services. And as part of this, they want to provide genetic testing. And I've been lucky enough to be involved in that. They've called me in, and I've sort of been consulting to them through my business to give them ideas on what I think people want and what practitioners want. And it is awesome because, there are so many different testing companies out there. And sometimes, as you would have heard at the conference and anyone that's heard me speak, I think Fit Genes is good because it's got so many inflammatory markers, but then maybe Smart DNA is good because it's got some more with some of the mental health stuff. And DNA Life has got stuff just on estrogen, if you want to know about estrogen detox. And then 23andMe, oh my goodness, it gives you so much. And 23andMe is amazing if you know what to pull out of it, because you get so much. But so there's all these different companies that offer these things. But now bioceuticals are coming up with something and have asked me to sort of, I guess, oversee or have some involvement in that. And I've been able to help encourage a lot of these SNPs and genes that personally that I want, but hopefully that other practitioners will love, because we're putting a lot more into this. And again, it's all based on evidence. And like I explained at the Bioceutical Symposium, which is how we began talking, the issue with 23andMe is that it's okay if you have someone that knows what genes to pull out, but a lot of the genes or the SNPs, those variations in there, are not based on robust or reliable evidence, and some people get misled by that. And a lot of practitioners don't have the time. We're all so busy, we're all trying to do things. You don't have hours and hours to go and look up the research and say, well, is this SNP, is this one evidence-based, is this one not? And so for that reason, I think that people can, get a bit misled by 23andMe, or maybe start to overemphasize some of these SNPs that really don't have hard evidence. Whereas with the companies here in Australia, they sort of have to go through that sort of TGA process, as far as I know, though I think I might be a bit confused about that. Someone was telling me the other day, nutrigenomics is not TGA, but I have read that it is, and I know that bioceuticals are going down that path of making sure that everything, is above board and based on research. But what we've been doing, and they've got a team of people that I've been lucky enough to work with, and as I said, sort of guide them on making sure we can get as many of these SNPs, and a lot that the competition doesn't have here in Australia, a lot that 23andMe do have, but we are evidence-based. And we're actually even coming up with, I hope this is going to come about, it should be, because it's going to be launched sort of mid-year, we're even coming up with a rating scale. So what pet practitioners can say, oh, wow, MTHFR has got heaps of research, we can put a lot of weight on that. But let's say now, okay, we know that the enzyme, or we know that this particular gene has got some research about the variation, but maybe it's not as strong. So yeah, there's an effect, but we want to look at it together with, say, serotonin levels or with COMT or and the other thing is we're trying to put things together, because like I always talk about, it's not a single SNP. We're trying to actually show ways of how these things work together. And on top of that, it's going to be quite interactive for the practitioners, and we're also offering some guidance about further testing, because what I say to everyone too with genetics is, remember, the genes are just about your risk or your predispositions. We're working out what are your genetic strengths and weaknesses, but we don't know if the genes are having an effect until we go and do other biochemical or functional or blood tests to find out, well, is MTHFR a problem for you? And then we look at your bloods or functional testing and we'll say, yes, it is a problem. You're low in folate, or we're looking at your methylmalonic acid. It shows that you've got a B12 deficiency. This is going to be a huge problem, and now we want to focus on that. So and along with the genetic testing, we've got suggested tests to help you really maximize the genes. And I think sometimes that's what practitioners are looking for. They want to know how to use this, what do I put it with, and and what are the other genes that work together. So
well, that's exactly what that's what we're looking for. Yeah. So I'm looking forward to hear more, and I'm hoping that down the road there's also something for digestive health, because what we're seeing as practitioners, our patients that are very much under a great deal of duress, and a lot of them are anxious, and obviously we would want to look more into neurotransmitters, but also, is there something going on that doesn't allow them to overcome these digestive disorders? And something I just read about FUT2 gene, and we're going to get a little bit more into it, because you talked about FUT2 being a part of the B12, I think it was, that you talked about. Yes. But what I learned was that those people that have this gene or variants in that gene would actually have a little bit more of a difficult time overcoming dysbiosis. Yes. So can you talk more about that?
Yeah. So and anyone that happened to just be at the ACNAM conference not last weekend, the weekend before, would have heard me talk about the FUT2 and the TCN2 and the MTR. So what I do is I try to sort of group all these SNPs, and we had a great case study of a little boy with autism who now goes to a mainstream school, and even the mum came along to talk. It was a really lovely case study we put together, but he had the FUT2, which is linked with B12 absorption and that gut microbiota that we'll talk about, and the TCN2, which is the vitamin B12 transporter, and then obviously the MTR, which uses the B12. So we can get a lot of information about the use, the absorption, and the transport of B12 from the genes. This is why I think the genes are such a good value. Sometimes when I say, oh, genes aren't a standalone test, people go, well, then why would you do it if you've got to go and do functional testing? Well, we want to know, is that person at risk of B12 deficiency? Is that person at risk of having low vitamin D? Is this something we're going to have to monitor and watch? And with the FUT2, so this is, it was actually discovered, the link with the B12 and the gut flora, through genome-wide association studies. So these are huge studies where they just go out and test, so many genes or so many genetic variations, and then see what it's associated with. And what they found is that particular gene variants of the FUT2 are significantly associated with just lower B12. And initially they're thinking, well, what's the link with vitamin B12? But we know that vitamin B12 is actually quite difficult to absorb in the gut. It's a huge molecule. It's got to bind to intrinsic factor, and then it's got to bind to other things, and obviously that transcobalamin, TCN2. So it's a difficult process to absorb. And of course, we also know, and I'm sure you know way more about this than me, is that gut microbiota, or that good and bad or commensal bacteria in the gut, is going to affect the absorption and the health of that gut lining, or that gut integrity. And the FUT2 is actually, it's associated, what we call it is a sort of secretor. Depending on the genotype, you're a secretor or a non-secretor. And then depending on these secretions in the gut lining, directly influences the gut bacteria. And there's research to show that particular genetic variations are associated with altered levels of the bifidobacterium and altered levels of B12. So the theory is that because of the FUT2 affecting these secretions, affecting that gut microbiota population, it therefore influences the ability to absorb B12. So we generally say FUTs linked with absorption, but it's not direct absorption. It's more to do with that, those healthy populations of bacteria in the gut. So FUT2 is something that, your particular audience will probably want to know about, because it does influence the gut bacteria, and it can help you. I mean, we often do stool testing when we want to know what's going on in the gut, but if your patient doesn't have the money, which, we're always trying to sort of work out what's the prioritize things. If they do have the FUT2, you might want to assume, or you might be able to assume, they're likely to have less of that bifidobacterium, and that might be a good probiotic that's going to help get them back on track, along with all the other things you need to do.
This is where I'm really excited about some of the PCR testing that's available now for microbiome, like uBiome and all that.
I just did that.
You'll have to wait 3 months to get your results. That's the only problem with that test. Takes forever.
If anyone jumps onto my Facebook, I don't know if it's still there. This was on the weekend, and it's now, Tuesday? I think this is Sunday. I got a link sent to me, and I used the code, and it was free. So if you use a particular code, it was for the first 1,000 kits. It might not be available now, but for the first 1,000 kits, and I did it on Sunday, you put in a particular code for the uBiome, and all you have to, the kit is free. You just have to pay for shipping, which was $19 US, which is about 27 Australian dollars. So I don't know if they've sold 1,000 since Sunday, but if they haven't, you might be able to get a free one. Everyone out there listening, I can't wait to find that out. Though I didn't know it was 3 months. I didn't know that.
Yeah, it takes 4, and if they're doing this promo, it might even take longer now. But I got that same. Somebody forwarded it to me, and it was like something on Reddit, and I was just like, you know what? I didn't know if it was like gonna lead me down to some dark internet hole, so I didn't I didn't really click on it.
It works.
That's good.
If you click on it, it went straight to the uBiome website, and then I bought it, and then I copied and pasted the code and put it in their little promo code thing, and it just took the $89 US away.
Wow,
it's obviously just for the standard one. It's not the one where you can do all 5 different areas. It's just the
you saved
one.
Yeah, you saved $89 US. It's great.
I know.
Okay, well, it'll be really interesting. So, I do use that a lot. And so in relation to FUT2, that might be really something to consider if you have a patient with that. So moving on from that, one of the things that I see often are people that have a lot of detoxification difficulties, especially with phase 1 or CYP of P450 systems. And something that I didn't know before, I learned this from Dr. Anderson when I went to, just recently I spoke in Chicago, and he also talked about SIBO and methylation issues. And he's sort of like, naturopathic royalty in America. So we do listen to what he says. And he talked about CYP enzyme system or the detoxification is actually really prevalent, more prevalent in the small intestine or in the intestinal tract than in the liver. So have you heard that? Is that correct?
I have to admit I didn't know that the cytochrome P450s, which are that huge family of phase 1 detox enzymes, was more prevalent in the gut than the liver. So I can't say if it's true or not true, but it also wouldn't surprise me, because we know that the gut is also a place of important detoxification. So when you're eating food, that has to then be detected. And, in the gut, it's like, well, is this good? Is this not? Do I have to detox it? Is it healthy? Am I going to absorb it? Is it a poison? So there is so much detoxification happening in the gut. And this is why, and again, I probably shouldn't even comment, it's so your area, but this is why if you've got issues, with the gut lining, that there are so many problems because you're not absorbing your nutrients, and you are going to get sort of these toxins, leaking through, causing oxidative stress, causing inflammation, probably going to affect methylation, and it's that sort of, that vicious circle. But the CYPs, or the cytochrome P450s, are very polymorphic, meaning they have lots of genetic variations, lots of polymorphisms, and these polymorphisms can either upregulate, so sort of speed the enzyme up, or slow it down. And the issue with a lot of people that have sensitivities and gut issues, and we see it with people with sort of IBS and things, is that they have a lot of genetic variations that upregulate, that speed up those cytochrome P450s, and your audience will probably understand, but some people sort of think initially, oh, that'd be a good thing. We're going to be breaking down our chemicals and toxins and these things in our body, but actually it's not a good thing, because the cytochrome P450s are phase one, and what they do is they produce these more interactive metabolites, and often these metabolites, or the products that the CYP P450s are producing, are more toxic than the original substance. So they're almost like a little red flag for the body to then come and say, cool, phase 2 enzymes need to come. We've got that conjugation, so add on a methyl group, add on a glutathione, add on some sort of sulfur thing. We've got all these different phase 2 pathways to neutralize these things and help us excrete them safely. So the cytochrome P450s are essential, but they can actually do harm when we have a lot of these sort of upregulations, particularly in those that have genes or SNPs within the phase 2 detox that slow down detox. And some of us, for those out there that do genetic testing, you would know that we have what's called insertions and deletions in some of the glutathione transferases. So the GST-M1 and T1, the glutathione transferase, those important phase 2 detox enzymes, some of us don't even have that. So when you're looking at the GSTs, we're not looking at a genetic variation. We're looking to see, do you have it? And there are a lot of people out there that have what's called deletions, and this means you still have that phase 2 detox. Of course, your body can still get rid of things, but it's a different pathway. It's slower, and those people need more support. So if you've got, faster CYP1s, you're producing heaps of these toxic metabolites, but you're not getting rid of them very well, then we have all these issues that practitioners like us have to deal with and manage and educate our patients on, what things to avoid. I mean, one good thing about doing genetic testing too is that if you've got particular genetic variations, you might then be able to say, well, you're going to be more sensitive to, say, benzenes, or you're going to be more sensitive to alcohol or cigarette smoke, or estrogens. When we're getting into estrogen metabolism, the cytochrome P450s, depending on what SNPs you have in what gene, you might be more likely to make more of these estrogen metabolites that are associated with things like breast cancer and a lot of health issues, estrogen dominance in females.
So I think that CYP1A1 and 1B1, as well as I think UGT is also involved in that, isn't it?
Yes.
In terms of detoxification and creation of sort of also catechol estrogens.
Yes, that's exactly right. And it's the catechol estrogens that can do the damage, and this is where COMT. So COMT is catechol O-methyltransferase, and COMT can break down these catechol estrogens. That's the safe pathway. So assuming you've got plenty of your B vitamins to support methylation, assuming you've got the magnesium, which binds with COMT, and there's actually evidence to show that depending on the level of magnesium will influence COMT activity, and that's really the whole purpose of sort of nutrigenomics. We want to try to give these nutrients to help support these enzymes. So COMT breaks down or adds a methyl group to these estrogens, neutralizing that, and then it can be cleared safely. But COMT also is involved in the breakdown and metabolism of dopamine, adrenaline, noradrenaline. So it's an important one that we definitely want to support, and we know that the genetic variation in COMT, similar to MTHFR, maybe not to the point of 70%, but the genetic variation in COMT does have a big impact on COMT activity.
So besides magnesium, are there any other support for COMT? Because that's one SNP I see a lot of, and a lot of people that have just relentless anxiety, which is sometimes independent of, SIBO has this gut-brain activity and issues that are unrelated to COMT. So I've seen cases of SIBO where anxiety was prevalent, and then as soon as SIBO is cleared, it's completely gone. Totally gone. So, and then you have those that have been anxious or because of this clogging up, as I usually say, of this COMT pathway or monoamine oxidase and this other downstream enzyme system that helps to clear excitatory neurotransmitters. So are there any other nutrients that you know that can really support these patients that have these particular SNPs?
Yes. So the magnesium we know is good just in general for calming it with the COMT, and also magnesium does so many things as well. We also know it can act as an antioxidant and things. But importantly, we have the B vitamins. So COMT is a methyltransferase, and we know that the methyl group has to be added or transferred to these neurotransmitters to help break them down. So if someone has a genetic variation in COMT that slows enzyme activity, what can happen is someone sort of gets, and we know this is associated with sort of stress and anxiety. Someone can get stressed out, and they have an increase in these neurotransmitters causing that stress and anxiety. And if you've got a genetic variation that slows that enzyme activity to break it down, they might feel stressed for longer or not be able to cope with that stress as well. So we want to make sure that we're supporting the methylation, because if they are getting stressed, they're going to be using up more of those methyl groups. So not only do we need it for gene expression and we need it for detox and all these things, your COMT is going to be using a lot of that up. So you want to make sure that you are looking at your B12, your folate, your B6, even your B2. And if you are going to support people that are, and it sounds like the patients you're working with these gut issues can be quite sensitive, you might want to do one step at a time. And I always start with the B12 and then think about, either the folate or the B6, depending on the patient, and then you sort of bring in the folate later, because you never want to just start with folate, because if anyone is low in any of these other B vitamins, you're going to get something called the folate trap. If you don't have the B12 and the other Bs, you start to get a buildup of something called 5-methyl tetrahydrofolate, and then everything slows down. The MTHFR slows down. You don't get the methylation side, and then you could actually possibly doing more harm than good.
And also, I completely agree with that. I think that oftentimes I see people that have been, for various reasons, they've been tested for MTHFR and they're heterozygous, let's say, for one of either 1298 or C677, and they've been put on huge amounts of active folate and have reactions to that. So, can you talk about why that happens besides methyl trapping? I mean, is there some element of speeding up cycles where downstream cycles have not been really adequately addressed? Is that what's happening?
Yeah, well, this actually comes back to you and all your amazing practitioners who work with gut health. At the end of the day, the gut really is where to start with things. And if you're going to go and give someone high amounts of things and they've got toxins and oxidative stress, you may not get the benefits. Number one, high dose, I think, for most things is not the answer. But I always say start with the gut. And for those that know me, you know that I work at a medical practice with a practitioner that focuses on the gut. So often we will sort of share patients. And you find that when you actually sort out the gut, people can tolerate a lot more things. However, the high dose folate, yes, gut's important, but sometimes the high dose folate too can sort of alter this methylation balance. And I think when you start playing with that methylation, you're going to get side effects. So I'm not one to give high doses. I mean, I do know that there is some research out there and some practitioners swear by giving high dose methylfolate for depression or anxiety. But I personally haven't seen that. I often get patients that have found me because they have had side effects, and now they're looking for someone because they've heard about methylation. They've tried to, support methylation, and actually it's ended up making them feel worse. So we always sort of strip everything back, start with one supplement at a time, start with, even lower doses, and then build up, have, depending what it is, a quarter or half a capsule for a few days or even a week, see how you feel, and then build up. Because sometimes when you give these high amounts, you're actually causing effects that you're not aware of, particularly things like B12. A lot of people that are chronically ill will react to methyl B12, and we know that oxidative stress, in particular peroxynitrate, can actually block MTR, that enzyme activity that requires B12. So in those cases, if your patient has serious oxidative stress, which is highly likely if they've got gut issues, think about Hydroxy B12, because hydroxy actually can act as an antioxidant as well, and then the body can either convert it into adenosine, which supports the mitochondria and the energy. So if they're having energy issues, it can go off that way, or it can be made into the methyl, and it's a little bit gentler. You're not sort of pushing these methyl groups into the body and sort of freaking it out, for lack of a better term.
That's really helpful. So would you always prefer hydroxy B12 in those that have potentially high oxidative stress?
Yes, always now. So when I started in the clinic, and anyone that did see the case study the other week at Acnum knows that I used methyl B12, and we got fantastic results. This little boy started speaking. It was just, he's now at a mainstream school, and the mother almost thought it was the methyl B12. However, she'd been working with a practitioner on the gut for, I reckon, a year. Plus, she'd been doing all sorts of behavioural therapy. It just happened to be the methyl B12, because I told you he had the FUT2, the TCN2, the MTR, MTR, all this stuff with B12. It was almost like a wonder drug. But I've since learned in that time, probably in 4 years in clinical practice, we had a couple of people react badly to the methyl B12, and they were, one was a child, one was an adult, people that had high oxidative stress. So anyone now that has high oxidative stress, we'll give the hydroxy and we don't get those reactions. And even in that, sometimes we give the, for those that need it, they'll even have like the B12 injections. And when we give the hydroxy, they are absolutely fine. They feel great. So now that is a bit of a sort of a rule of thumb for us in the clinic, that if we have any signs of oxidative stress, we're not going to give the methyls. We're going to start with the hydroxy B12. And then go from there. And then they might have supplements down the track, B6, maybe some P5P, maybe a little bit of folate, maybe a bit of folinic, a bit of methyl, but definitely starting with that hydroxy, and always starting with the gut, which I'm sure you and your listeners are happy to hear.
Very happy to hear. Just as a sideline, do you test for oxidative stress, or do you just do how do you test for it?
So I am a scientist, not a medical doctor. And mind you, I think the medical doctors I work with do a lot of testing too, but I guess this is the way that I was brought up. Everything is, test and look for things. So I do a lot of testing. The only thing that stops me from doing testing is when the patients can't afford it. Then we might start to sort of try to make a plan of what's the most prioritized things. But oxidative stress, you can, if someone's looking at hormones now, you can do the Dutch testing, and you just get oxidative stress with that. If you wanted to do some bloods, there is a lab. I can't think it off the top of my head. I actually presented it at the Bioceuticals. You might have the information, but I can get that to you. There's a lab in Sydney. Patients have to pay, but it's not too expensive. It's about $80, and they can do blood, hydroperoxide, total antioxidant capacity, and I think that will allow you to find out, well, do they have the oxidative stress, which is represented through the hydroperoxide, but also what's their antioxidant capacity like, and we can order that as practitioners, health practitioners, and then the patients will just pay. It's not on Medicare anyway, so it doesn't matter if they see a GP or us. Also, you can just do the urine, 8-hydroxyguanosine. That is a good marker. And I don't often use it, but again, if you're really struggling with finances, I'm now starting to tend toward doing possibly some more hair analysis. It's not something that I used to do, but if someone's got, you're looking at that hair analysis, they're obviously telling you about their symptoms as well, but they've got issues with heavy metals and things like that. No doubt they're going to have oxidative stress. So I do try to test for it specifically, but if for some reason you can't, often you can put some of these symptoms together. They've got high mercury, or even, all the gut things. I don't think we've had anyone with serious gut symptoms not have oxidative stress, because, you've got that leaky gut. You're getting these things into the bloodstream. The body's reacting. You get all this, all these cytokines, this immune response and the inflammation, and then that inflammation then sort of feeds into the oxidative stress as well. So whenever you see inflammation and oxidative stress, they're generally hand in hand.
Totally agree. I do see that a lot. And just an aside, the Dutch profile of the dried urine, I think, what is it? Dried urine total hormone something. But it's
Hormone. Yeah, total
something.
TUTCH.
Yeah.
Comprehensive
Hormones. There we got
worked it out. Yeah.
They just added the 8-hydroxyguanosine, right? So that
and
is.
I don't think the price hasn't gone up to my knowledge, because I'm just back in clinic now, and I just ordered one yesterday, and the prices were the same. So anyone that's doing Dutch testing, you'll now get that 8-hydroxyguanosine, which is a very good marker. And even back when I was in the lab, because I used to measure, part of what I was doing aside from the nutrigenomics was DNA damage. So we would look at DNA damage markers as well as the 8-hydroxyguanosine.
Because isn't that, I thought that it was a marker for DNA damage, the 8-hydroxyguanosine.
There's actually a lot, because I sort of come from that fertility, pregnancy, health. There's a lot with sort of sperm DNA damage and 8-hydroxyguanosine. So it is, I guess, an indirect measure of DNA damage and oxidative stress. So when we were looking at DNA damage, we're actually really looking at DNA damage. I used to sort of look at, culture cells, like people's blood, and then culture them and look at them under the microscope, and we can actually see like breaks in the chromosome, or we can see something called nuclear buds. Like we can actually see the damage in the nucleus. But the 8-hydroxyguanosine is like a metabolite that gives you an indication of that things are not good. But it is a direct marker of a marker of oxidative stress and DNA damage.
Great. And for our overseas listeners, we have a lot of American listeners. I think Doctor's Data does offer this as a single test as well. So just look out for that. Now,
I think there's quite a few. I think there's quite a lot that offer the 8-hydroxyguanosine. So that's something that, it's something that most of us could do.
Great advice. Now, you mentioned obviously absorption of endotoxins and leaky. Well, you didn't say it, but I sort of deduced that from when we have digestive problems, oxidative damage, leaky gut, there is just a lot more absorption of endotoxicities, such as lipopolysaccharide. Does that affect certain? Does it affect certain genes or so? Because I've heard that LPS can affect monoamine oxidase, and it can, I think, affect a couple of other genes that code for particular enzymes that are also important in this conversation that we're having.
I think that there are lots and lots of genes that are affected by these. I couldn't tell you all of them. At one point, we were sending blood to a lab in Germany. I'm trying to think of the name. I can let you know the name, but the truth is they're not taking samples from anywhere out of Europe anymore. They got too busy. But we used to send blood for our really complicated patients. We would send blood off Acumen. It was called Acumen. So we would, if you've heard of Acumen, I don't know if you're from the Netherlands, is that right? Do you hear about Acumen over in Europe?
I'm actually originally from Germany, but I left a long time ago. So, but I have heard of that lab. Yeah.
Yeah, so Acumen, we could actually look at the effects of certain chemicals or products, sitting on DNA and almost sort of switching them off. And often you would see things like, it'd be on detox enzymes, or things like, MAO and COMT that are sort of breaking down neurotransmitters. So for some reason, these tend to be targets. I have no idea why, but you are completely right that when these things are passing through the gut, not only are they causing oxidative stress, but they actually can be switching off or, affecting genes and how they're expressed, and also then influence sort of the methylation, because the body has to work so much harder to clear these things. It's going to be drawing on that methylation cycle, drawing on that glutathione, and if you see anyone that has a low homocysteine, that's going to ring alarm bells. Back when I was a scientist, we always thought high homocysteine is bad, but now that I work in this area, a lot of these patients that are unwell have low, and that's because the homocysteine is being pulled down that transsulfuration pathway to make glutathione, but also pulled off to make methylation for detox. So anyone that's got detox issues and gut issues is likely to have a low homocysteine because the body has this sort of increased need. And then when you've got the issues with the methylation, then that can affect COMT, even if things like COMT weren't targeted by some of these things passing through the gut, you're going to get reduced activity because the methyl groups just aren't there.
So how low is a low homocysteine in your eyes?
I think the lowest I've seen is about 2.
Oh, really?
That's usually,
wow.
Yeah, the autistic children. But, once you sort of, 4 and under, even 5, a bit of more. 5 sometimes can be a healthy person that's just having a lot of B vitamins, so they're just really pushing that methylation cycle. But 4 and under, we want to say, all right, there's something going on here with detox. Is there something in the liver? Is there something in the gut? Like, is there something, if there's no obvious symptoms too, you want to start investigating all of that detox. And we mentioned briefly some of the, cytochrome P450s and the glutathione peroxidases, but we also, when you do genetic testing, can look at MN-SOD, so superoxide dismutase. That is a very powerful antioxidant in the body, and that's sort of step 1 in that antioxidant cascade. So it breaks down superoxide, which is a really nasty free radical and can cause oxidative stress, into hydrogen peroxide, and then you've got catalase and glutathione peroxidase that break that down. So often people with gut issues will have oxidative stress, and some people are genetically better at dealing with that oxidative stress and others aren't. So if you have a patient, and I do frequently see this, that have variations in superoxide dismutase, catalase, and glutathione peroxidase, we already know genetically you're predisposed. You're likely to have high oxidative stress. We really need to support this. And the superoxide dismutase, you want to investigate manganese, because manganese is what works with that, and that's particularly for the mitochondria too. So anyone having energy issues and oxidative stress that can be affecting fatigue. And then with the GPX1 or glutathione peroxidase, that's what we call a selenoprotein. So it needs selenium. And then catalase has the cofactor iron, but I haven't seen any research showing that iron levels impact its enzyme activity. But no doubt, if you're low in iron, you're not going to be able to get that adequate enzyme activity. So the antioxidant enzymes, along with the detox phase 1 and phase 2 and the methylation, are often what I look at as a whole when we're trying to work out what's going on with that person, and also trying to identify where you start, what is the area, because we obviously don't want to bombard them with just heaps and heaps of supplements and everything at once. It's sort of a step-by-step process to get people well. So we'll sort of look and say, well, if it is detox or oxidative stress that is an issue and genetically is a problem, we might want to start there, even though methylation is so important. And you mentioned that's where my research was, and that's what I do. It's obviously not the only thing going on, and it's not always the best thing to start with. You might want to start with, like I said, with the gut, but then also supporting oxidative stress. So making sure that they're getting adequate antioxidants in the diet, making sure they've got plenty of selenium to bind with this antioxidant enzyme, the manganese, etc. And obviously always, I'm sure all of your practitioners do it, but the zinc and the magnesium, they just seem to help with everything.
Yeah, pretty much. So, but like you mentioned SOD, catalase, and glutathione peroxidase. I know that the 23andMe does. I've seen SOD, but I haven't seen catalase or GPX1. So are they on that panel, or do I have to go to a different lab, or?
No, I think, why don't you leave it with me? I just did my son's 23andMe actually. So I've got the recent one, because they do sometimes change their chip. So, back in the early days, they used to do heaps of HLAs, which was great for my autoimmune patients. They don't have all those anymore. But I just got my results from my son last week. So I'll have a look. I'm pretty sure GPX was there, but not sure if I saw catalase, but I can get back to you so your practitioners know. And on that, I think you heard me talking at Bioceuticals, and I was saying I don't recommend 23andMe. The truth of the matter is, if I talk frankly, you are going to get more from 23andMe. The reason I steer away from it for some of my patients and practitioners that are moving into the area is that when you use these companies here in Australia, or even overseas companies that are distributed here in Australia, you get a report that patient can take away that's got an explanation, or it's got some pictures, and it's in a way that helps the practitioner explain it, but also the patient's got something to help them, I guess, say, oh, okay, well, there's some stuff with methylation, or there's some stuff with inflammation. Whereas with the 23andMe, depending on the practitioner, it might be difficult to hand over something or explain things. In saying that, I work with a practitioner who loves 23andMe, and what she does is she doesn't go into the depth that I do. I guess because I have a genetics background, I like to explain. I tell them what the strengths and the weaknesses. I'll be like, you look at this, you've got great antioxidant enzymes. Obviously, it's a lifestyle thing, so let's sort that out, because genetically, you're good. But I know some practitioners will just get 23andMe and just pull out a few things they know and then push it to the side, whereas I spend a lot of time on the genetics. So if I did an Australian company, I can actually go through everything. and explain all of those things. And I guess not everyone, but most patients, I think, appreciate that. The kind of patients that are doing testing or that are coming to see me actually want to know. They're educated people that have been doing reading on the internet. They want to understand these things. So I go into quite a bit of detail with the genetics.
I do use 23andMe, and only because it's cheap, right?
Yeah.
So the big drawback with some of the more sort of glossy reports that you get, which are wonderful from some of the other labs, but
Yes.
the patient has to pay $500-$600.
Well,
and
guess what? I know. I've actually spoken to bioceuticals, and I can't say the price in case it's not confirmed, but I was blown away at the cost. So they are not.
Okay.
Should be careful what I say. This is just my opinion. From what I understand and what I've been told, they're not out to make big money from this. It's to support. They want to move into this sort of clinical service and personalised medicine. They see this as the way of the future. So they're offering it, and I was shocked at the price. And if it is what they've told me it is, it's so affordable. And I can tell you firsthand the amount of effort and research that has gone into making sure that these are evidence-based, and the way that we're trying to represent it to help the practitioner use it. So watch this space. Obviously, I'm biased because I've been working in it, so I think it's amazing. But we really are trying to make it more affordable and accessible, because that is a concern not just for practitioners but the patients. I mean, that's my biggest issue. Is the money that it costs to go and to dig this deep. It can cost a fortune doing your organic acids and your Dutch and your genetics and other functional testing. It's ongoing.
Yep, I can completely relate and attest to that. My patients are often extremely. They've been referred to me. They've already done a lot of testing, and sometimes, the reason I also do the 23andMe is because you do get a lot of information. But I just want to echo what you just said and just reiterate to the practitioners out there that it's really important that you do get more training on methylation. I've done a lot of training, and I feel like I'm still just peeking over the fence in some ways. And I've done days and days and days of training on it from different sources, just because there is a lot of information out there. And so I do encourage practitioners to not just take little sound bites from what we talked about, but really use it as a platform to do your own research and to find a place in your country where you can get some education around that. So that will be wonderful. I'm hoping you're developing something, Denise.
I was just about to say, if you don't mind me promoting myself.
Please
time
do.
has been an issue. I obviously have a little baby, but my amazing husband has resigned to look after the kids because I've been so busy. But that is the biggest thing I'm always asked about, education. And sometimes people that are providing education might want to sell a product. So I've actually just had a huge conversation with one of the doctors I work with, a medical doctor at the practice, and we're getting it from all the patients and practitioners. We're going to design some sort of education. We're going to run a workshop at the clinic in Melbourne and then see how it goes. And then based on the feedback, I'll design something that's online. So I want to start putting things together pretty casual. I'm not out there to make heaps of money from it. It'll be something just to start to educate people and get feedback, and then maybe down the track, and not just on my own, get in other experts, because yes, I know about methylation and genetics, but it's so much bigger than that. If you want to talk about gut, we can get you in, Nirella, and put something together which is about clinical applications. So yes, education, but how do you use this in the clinic? Why would you use this test? What's the difference between this one? And again, like I always do, not trying to say one test is better than the other, but giving people that objective feedback so that they can take that and then go, cool, well, I can go next week into clinic and order this test and this test because I know what I'll get from it and I know how to interpret that. So it's obviously big picture down the road, but I am going to start with some smaller education and webinars. As I said, some face-to-face, which I've been doing face-to-face stuff before I was on maternity leave at the clinic. We were doing quite a lot of that and doing some webinars, but I am going to start moving that into sort of on an online presence. So hopefully people will appreciate that. And I'm also so open to feedback from you and others on, tell me, yes, I understand you, or no, you're too scientific, or you didn't cover this, so that we can all learn together. Because
Fabulous. I think it would be great. I'd be very interested in that.
Yeah, I think for us to get the success that we do, it's about really networking and also referring back and forth, because we all have our specialty, as you do and as I do. But I think when we really do truly have those chronically ill people or people that aren't getting well or have things, sometimes it's nice to be able to leverage off someone else's skills or put all this stuff together, because it's the combined effort that gets truly those amazing results.
So absolutely. So before, we've got about 10 minutes left. So I just want to briefly touch on the transsulfation pathway, getting back into the whole conundrum. So this is where cystathione going down to beta synthase, CBS pathway going down towards glutathione. What can go wrong there? What cannot go wrong there?
Well, I might open a bit of a can of worms here, because I might disappoint some people. And I have to admit, sometimes I do disappoint people because I am evidence-based. I'm looking for the research. And that doesn't mean that I know all research, but I am trying to keep on top of this. The CBS is quite a controversial SNP. So cystathione beta synthase is the first step in the transsulfuration pathway. It turns homocysteine into cystathionine, then into cysteine, then it can come down to sort of taurine or glutathione. So it's hugely important in detoxification. Now, the genetic variation that is often the one with the most research that is often tested for is an upregulation, which means it speeds up enzyme activity. If you look at the research, people report that as a good thing, because it says, well, if you're speeding up activity, you're going to be pulling homocysteine down the transsulfuration pathway. You've got lower homocysteine, and we know that homocysteine is nasty. Homocysteine causes endothelial cell damage, vascular things, pregnancy complication, dementia, Alzheimer's. Homocysteine's a nasty one. But I think the kind of patients that we see are not the typical, and they often have the low homocysteine because they've got this huge requirement for glutathione. So if you've got low homocysteine and your CBS is sped up, that could be a bad thing, because you're going to pull it down. Also, when homocysteine is pulled down that transsulfuration pathway, you get the production of gases like vasodilation. So often you might find that your patients that do have detox issues too have low blood pressure because of that vasodilation. But what I read about that I haven't found evidence for, and believe me, if there's some listeners out there that have the evidence, I'm down for you to share that with me. I'd love to know. But what I can't find the evidence for, that a lot of people talk about, is sort of these sensitivities with sulfur. They'll say if you've got the CBS, upregulation, then what happens is you're going to get all this sulfur, because we're talking about that sort of vasodilation, the sulfur gas, and you can't tolerate sulfur foods. I don't think there is evidence for that. Yes, I work with people that can't tolerate some of the sulfur foods, but I think it gets back to more gut than CBS. So if someone has CBS and they can tolerate foods, the last thing I'm going to do is cut out all sulfur foods. I think that I think it might have come, some of it, from Amy Yasko, and she does some great work. She's a scientist like me and had children with autism, and I think a lot of her stuff might be clinical observations, like with a lot of us. But the evidence, the actual science to show that when you've got CBS, that means that you are sensitive to sulfurs, I don't think that evidence is there, and I think it's more to do with the gut. The other thing is, if you do 23andMe, yes, there's less evidence, but a lot of those other SNPs actually slow down CBS. So sometimes just having one SNP doesn't really give you the whole picture. And I think when it comes to CBS, if, for example, you wanted to speed up activity, you want to give B6. It's the cofactor. So you want to give your P5P, your vitamin B6. That's going to help pull homocysteine down to transsulfuration and glutathione if the homocysteine isn't low. So if it's not low, give the B6 to help support glutathione. But aside from that, I think we have a lot to learn about CBS. I am looking forward to looking a little bit more into the 23andMe, because I think, they've got things like SUOX and other genes. But I have to do a bit of reading. I know that the genetic variations can have some impact, but I'm just not sure about the significance or the relevance in the big picture, unlike things like COMT or MTHFR or, superoxide dismutase, where we know the gene actually has a significant impact. I think some of these we're not all over it yet. So the good thing is, like what you would have seen at the symposium, there's so much research coming out. The last couple of talks I've given so many papers from 2017. The research is happening. This is like a hot area. But yeah, maybe that could be a topic that we can do another one on CBS, or I can do a webinar or something and try to really pull that apart, because I have looked very hard for a lot of this research, and it's just at this point in time, I don't believe it's there.
Yes, I agree. There's a lot of controversy around what is out there. And you mentioned Dr. Amy Asko. I do think that there are a lot of opposing thoughts to that. And
yes.
I do agree that when I do see issues with sulfites, it's more the sulfite oxidase pathway, not necessarily CBS pathway, as you mentioned.
Yes.
So that's more, we know that exists, that sulfite oxidase turns sulfite into sulfate, which can then be excreted, and that's a molybdenum-dependent pathway. And we do know people, if they're sensitive, truly sensitive due to some variation in that gene, then sometimes people really do improve with molybdenum. Rather than, with CBS and taurine, there's all this interplay that I don't even want to get into. I just was curious what you were going to say, because I know there's a lot of confusion around it,
Well, I didn't really give much today, but hopefully I explained in a long fashion
yes, you did.
That the evidence isn't there. And as I said, if someone listening has some great research, not just stuff you've read on the internet, but has some research, please send it to me, because I always say I don't know everything and we're all learning together. But at this point in time, I think don't put as much weight on it. I mean, I even read something, I probably shouldn't mention the company. They're a great company who does all this meal planning stuff. Sometimes we use them at the medical practice. But, they sent out an email and said, if you have CBS, you need to cut all of these sulfur foods. I thought, wow, you're not even talking about any symptoms. You're just basing it all on a gene. So some people have really taken it to the extreme, as a word of caution. Don't do that with any genes. We don't want to put all the weight on just the gene or just the SNP. Like I've said, look at the other SNPs, look at the diet, look at the lifestyle, look at the symptoms and other testing if they can afford it.
Excellent advice. And just if I can just sort of summarize that, well, maybe not summarize, but just so that I can kind of understand some of the really major points that you've made in this podcast is like, we have the genetic testing available, but then following up with which is sort of like the scaffolding or the blueprint, and then following up with potential actual levels of S-adenosylmethionine or folic acid will give you actually the function of these genes or the activity of these genes. So not just looking necessarily at the SNP, but looking at what does it actually mean for that particular patient. I think that was also a really important point. There were so many excellent little clinical pearls that you have thrown out here. I'm really hoping that all of this will be very useful to our clinicians out there, which I'm sure it will be. And thank you so much for your time, Denise. And I really look forward to seeing what you create with Bioceuticals, and look forward to these panels, and hope that you come back to us on the podcast when some of this is really well established and you can report back as to what people are saying out there about
maybe we can do some case studies, or even if you use it, we can have an open, objective conversation about if it's working, and if it's not, what we need to do. I mean, one thing I'm loving about Bioceuticals is the people here are really open to my suggestions and working together, and there are other practitioners that they've pulled in as well that do genetics, and they're really listening to what we want, and we're trying to develop something that is a bit more user-friendly and affordable, as I said. So it'd be great if practitioners do use it, or you and I, we can have a conversation to sort of say, yeah, it's great, or actually we've forgotten this, or this bit's confusing, and work with them to try to develop and deliver something that we can use. Because I think one of the biggest issues with genetics is that people, obviously have a degree in genetics, and I've done nutrigenomics and a PhD and a postdoc, and people don't have that kind of education, and sometimes people are a bit scared to include it, and I do think it does add so much value. So if we can have something that is user-friendly and has the support, the good thing about Bioceuticals is, they're a large company, and I'm doing training for their tech support. They want to know, obviously you can't answer everything over the phone, but they are in the process too of making sure there's education and there's tech support to answer your calls. So hopefully it's as good as I am making out it is.
A girl can dream. A girl can dream.
And if not, just let me know and I'll send it back to them.
Yeah, I mean, look, let's also, we are also aware that it is a company and it is there to make money. So
yes,
that is a reality. So, but I am look forward to seeing what they have to offer. And if again, we have a lot of overseas practitioners, so please find labs in your country that can offer some of these panels so that you can really understand your patient's plight a little more. I guess is also the message here.
Absolutely.
Any last minute parting thoughts, Denise?
No, thank you for inviting me, and thank you for being interested in all of this. I am keen to come back when we, can talk about some of the use of these testing in application, and if I do get some of this stuff happening on my own website, then I can invite you to come and talk to the genetics people on SIBO. So
I would love to. I'd love to. I can talk about it in my sleep. I'm sure I do, actually. Great. Thank you so much for your time today.
No problem. Thank you. Bye.
Thank you for listening to the SIBO Doctor podcast. We hope you found the information in this episode useful in the treatment of your SIBO patients. Head over to our sponsor, SIBOtest.com, an online testing service for your patients and home of the Practitioner Education Portal. Tune in again for another episode of the SIBO Doctor podcast. Thanks again for listening.